Hello everyone and welcome back to another 1014 BPS example calculations tutorial. So my name is Brock Grant and let's get stuck into a few of our week 3 questions. So our first question here is asking us to calculate the isoelectric point of two amino acids, alanine and glutamic acid. So let us start off with alanine. The first thing that we need to do when we're looking at calculating the isoelectric point is to remind ourselves what it is we're even trying to calculate. So the isoelectric point tells us the pH in which our molecule is neutral in charge. In other words, a zwitterion. So the first thing I would recommend doing in this instance, especially as we're being introduced to the topic, is let's draw out our amino acid. Now, how I've drawn alanine here is typically how it would look at a pH of 7. Now, we're also given some values here. We are given a pK1 and pK2. So our pK1, we can see it's around 2.34. So considering that the R group for alanine down here is non-ionizable, it is safe for me to say that the pK1 is going to be for our carbox and the pK2 is going to be for our quaternary ammonium group. So how do we calculate the isoelectric point? In this instance, we simply take the two pKa values, add them together, and divide it by 2. Which in this case is going to equal to 5.97. Now, let us move on and have a look at our next amino acid, glutamic acid. Now, for glutamic acid, what we see here are 3 different pKa values. Why is that? Well, the R group down here is ionizable, so we need to take that into account. So we have 3 pKa values. We've got 2.19, 9.67, and 4.25. So how do we go about calculating this? Well, as I said, it's important for us to remind ourselves what we are trying to do here. So calculating the isoelectric point means trying to find the pH in which this molecule is neutral in charge. So if I was to just throw out a few random pH values, so if we look at, say, a pH of 1, what that would do is that we can see that sort of all 3 of our groups here are going to be ionized. They're going to be protonated. Now, if we change the pH to, say, a pH of 7, then what we can see by looking at our pKa values, which are 2.19, 9.67, and 4.25. So what this tells me is that in order in which these groups are going to be deprotonated are going to be 1, 2, and 3. So overall, looking at this amino acid at a pH of 1, it's going to have a charge of +1, 0, and 0. So the overall charge is going to be +1. Okay, so what happens if we increase the pH slightly, if we make it more alkaline? So if we push this to around a 3, what's going to happen is our carboxylic acid group on the side here is going to become deprotonated. Now, what we see is a charge of -1, +1, and 0. Okay, now we're getting somewhere. So to calculate the isoelectric point, what I want to do is take the pH in which I know it's going to be neutral. So looking at this, in order for the next group here to lose a hydrogen will be around 0.25. So what I need to do then is take the average of this one and this one. Why? Because I know the isoelectric point is being controlled by whether this group or this group is protonated or deprotonated. I do not have to look at the quaternary ammonium group here simply because by the time we get to a pH in which this quaternary ammonium group can be deprotonated, it's too late. We've completely overshot our isoelectric point because of these 2 groups here. So it's going to be 2.1 plus 4.25 divided by 2, which is going to equal to 3.22. And that's how we calculate the isoelectric point.